Good evening, everyone, and welcome to tonight's Be Curious LATES event. Although slightly less late than it was supposed to be, because we moved this event earlier since it's coming home. And by it, I mean our speakers coming into your homes by the power of the internet to talk about shaping clinical futures. My name is Alex Holmes, and I'll be your host for tonight. I'm a PhD student in the Faculty of Biological Sciences. I'd love to know who all of you watching are. Why not tell me where you're tuning in from in the chat on YouTube? This evening's event is part of Be Curious, which is the University of Leeds' annual Research Open event, where we invite you to take a peek behind the scenes of what goes on inside a university. We'll be going live with Be Curious activities and events to explore everything from the world-leading to the wondrous, right up until the 15th of July. So be sure to check out the rest of the programme after this evening's event. You're at one of the LATES events, which is for adults, but we also have hands-on activities for families after school each day. For example, yesterday I made a stethoscope. So back to tonight. We'll be exploring how research can influence our health and well-being and change NHS practice, and maybe even lead us to treatments of the future, with a bit of help from a fantastic lineup of 3 researchers. Now, each speaker has just 10 minutes to tell you about an area of their research. If you have a question for a speaker, just post it in the chat, which you should be able to see to your right if you're viewing this event in YouTube. If not, we'd recommend clicking the option to view the event in YouTube to get the best experience. Now, our moderator team, Jane and Jacob, will be keeping an eye on the chat all evening. So let's try and keep them on their toes. Let us know what you think and what you want to know all night. Now, we have 3 speakers. We have Eileen Tan, Kara-Louise Royle, and Nick Orsi. And first up, we're going to welcome on Eileen Tan. Let's bring her up onto the stage with me. Hi, Eileen.
Hello, Alex.
Let you take it away.
Thank you. So I hope you can see my screen, and technology is kicking off nicely, hopefully any moment. There you go. So my name is Eileen Tan, and I'm a rheumatologist and an associate professor at the university. As a rheumatologist, what I do is I try to help patients with joint pain and aches in their body to make them feel better. So for the next few minutes, I'm going to talk about how exercise is good for joint health, and to elaborate on that, show you some of the research that I have been doing. You may have seen in sort of media where there's a lot of questions being posed, such as, is running bad for your joints? Which are very common questions I get asked by my patients. They also ask questions like, will continuing to run make my knees wear out faster? Or indeed, should I keep on running if my knees hurt? But in actual fact, the short answer to those questions is running may actually be good for your knees, and why running won't actually ruin your knees, or it doesn't even wreck your knees. So I'm going to give you a flavor of how research can actually confirm these statements. My research involved a scanning technique called magnetic resonance imaging, or MRI. This is a special scan that uses strong magnetic fields in order to visualize the inside of the body. So within the research, I was particularly interested in looking at the muscles in the human body. One of the largest muscles that could be scanned quite easily using MRI scan is the leg muscle, or specifically the thigh muscles. So here you see somebody lying down with what we call a cage or a coil around the upper thigh muscles, so that we can focus on that part of the body within the scan. Now I'm going to show you some images of what we can see. So here are 3 MRI scans of the thigh muscle of different individuals. And the first image on the edge of the screen shows a 45-year-old healthy female muscle of the upper leg. The middle image is one of an 83-year-old healthy male, which shows some fatty, streaky, fatty infiltration that is associated with healthy aging. The last image you can see shows a lot of white streaky areas that shows that there is a lot of what's called fatty infiltration in the MRI scan of this 60-year-old male with a condition that affects inflammation of the muscle called myositis. So I was particularly interested to see what happens in the muscle of a group of patients with a type of arthritis called rheumatoid arthritis. This is a very common inflammatory arthritis. And this is Matthew Farrow, one of the PhD students who successfully did this project, scanning healthy control or healthy individuals compared with patients who had rheumatoid arthritis. So these are their thigh muscles. And you can see the difference straight away, that the rheumatoid patients have streaky white areas within the muscles. So in the scientific format, this is a graph that represents the amount of fat fraction or fatty infiltration in the muscles. And if you compare the bars, the healthy individuals are the blue bars, and all the other colored bars represent rheumatoid arthritis patients at different stages of their condition, including those that are recently diagnosed who has yet to be treated, those who are on treatment but continue to have symptoms, and those on treatment who feel absolutely well from the arthritis point of view. What you can see here is that in all the three groups of the rheumatoid patients, their muscles have significantly more fatty streaks on the MRI scan compared to the healthy individuals. So Matthew also assessed the strength of these rheumatoid patients, and similar trend of results were shown in that when the three groups of rheumatoid patients were compared to the healthy individuals, their muscles were weaker compared to people who were healthy with no rheumatoid arthritis. At the university, we often combine our research with other people as well, because we thrive on the expertise of other individuals. And here is a research we collaborated with our neighbor working at Leeds back at university. And this is Chris Bolchin, who did a PhD in looking at the feasibility of different types of exercise in people with early rheumatoid arthritis. What he did was he took patients and allowed them to do different forms of exercises that included moderate and high intensity cycling, as well as strength exercises, which are low and high intensity resistance exercises. And all participants then go through all these exercises compared to a period of stationary, no exercise periods over a 6-week period. And what he found was fascinating. All the participants completed all the exercises, but none of the exercise actually increased joint pain. So they didn't have any more joint pain doing the exercises. In addition, they all enjoy the exercises, which means that there is significant motivation to allow them to continue with the exercises. And this is consistent with what is encouraged in the population with people with arthritis. Here's a website from the Faculty of Sport and Exercise Medicine that on their website is highlighted in big letters, suggesting that exercise does not actually cause osteoarthritis. And if anything, exercise can actually help prevent and treat osteoarthritis. And for that reason, as a rheumatologist, I often recommend exercise to my patients because of the benefit it has for their joint health. And not only do they have to run, they can play football or any other sports that they enjoy. Any physical activity is better than nothing in terms of helping improve the muscle strength and the joint health. In the current climate, exercise is even more important because there's increasing evidence from research that suggests exercise can be good medicine because it improves our immunity, particularly fighting against infections. And if you don't feel like running is your sport or football is your sport, here are 21 exercises you do not need any equipment for, which can be done indoors or outdoors. Sweatbands are optional, but these are using your body weight, so you can do them anywhere, anytime. So I hope I've given you a flavor of the type of research that we have done at the university that shows that muscle health is important for joint health and exercise is important for both of them. Thank you.
That was so interesting, Eileen. I know we promised all the people watching a look inside the universities, but I wasn't quite expecting a look inside the human body. Um, there are a couple questions in the chat, but I'm host, so I'm going to be kind of selfish and ask one of myself. So there's increased fat in these thighs from patients with arthritis. Is this because of the reduced exercise, or is this something to do with arthritis itself?
So if you remember one of the slides that showed three MRI images of the thigh muscles, the middle image that showed the older person with the fatty, streaky muscles, that's a healthy individual. So therefore, the fatty changes in the muscle goes with age. However, we also know that probably exercise does either reverse or reduce the amount of fattiness in the muscle. And we also know that muscle with less fat is actually healthier. So it's not necessarily due to the arthritis. It's probably is due to the fact that with pain, people move less and therefore more sedentary, less exercise, and hence the changes are seen in the muscles.
Yeah, a combination of different things, whether that's pain, lack of exercise, all those things. Okay, that's really interesting. We do have a question also from the audience at home. So they really like the idea of prescribing exercise, but they wonder whether you have to sometimes do quite a lot of work convincing your patients to get on board with exercising.
I guess sometimes yes. I think it does depend on the individual, and it's individual cases that determines what level of exercise is suitable. Exercise could be as maybe as simple as walking. However, some people cannot walk, so they may be in a wheelchair. So we need to think of exercise that are suitable for them in a wheelchair. So I think it's very individual based. For my patients, because I know them, I know what level of exercise may be suitable for them.
That's really good, actually. Yeah, when you have that kind of personal relationship, you know what they would be interested and enjoy. No, I like that. Thank you, Jenny, for that question. That was really good. One final question then from me. You kind of show the 21 exercises. Which ones are your favorite to do?
Oh, well, well, actually, all of them. I think, I don't know if anybody does because they are so fun. And I think the importance of doing a variety of exercise as well is that you work on different muscles. So if you always do just the one exercise, you are going to neglect the other bits of the exercise. So therefore, a variety of exercise is probably very important.
And that makes sense, since arthritis can, be all your joints. Okay, well, thank you so much, Eileen. That was really interesting. So next up, we have Kara-Louise Royle. So we'll bring her up onto the stage with me as well. Hello, Kara-Louise. How are you?
Hi, I'm good, thanks.
Fantastic. Well, I'll let you take it away then.
Thank you. Okay, so I'll just get my slides up. So hi everyone, I'm Kara-Louise Royle and I'm a statistician at the Leeds Institute of Clinical Trials Research, which hosts Leeds Clinical Trials Unit. Now, over the past year, there have been a number of references to clinical trials in the media, a lot more than before 2020, and you may have not heard of them before. In the next 10 minutes, I'm hoping to give you a brief introduction to clinical trials research and tell you about the different ways you could get involved. To start off with, what actually are clinical trials? Well, it all starts with a question about a specific health condition that we want to know the answer to. For example, we may want to know what is the right amount of chemotherapy to give a patient with cancer, or whether a new medicine for IBS is better, worse, or no different to what is currently being used. However, it's not all about drugs. At Leeds Clinical Trials Unit, we also look into questions which compare different types of surgery and consider how lifestyle changes such as diet and exercise can affect a person's health. Now, just to introduce some terminology for the rest of the presentation, we usually call what we're looking into, so potentially the new thing, the experimental intervention, and what's currently used, the control intervention. But how does a trial actually work? Well, in general, there are 5 key stages to a clinical trial. First off is information and consent. Before anyone takes part in a clinical trial, they are given information about what will happen if they choose to take part. They can then agree or not agree to join the clinical trial, and this is called consent. However, because we do not know whether the experimental intervention is safe for everyone, There are generally a set of rules about who can take part in a clinical trial, and these are called eligibility criteria. Next up is baseline characteristics. Before we give anyone their intervention, we need some information about the types of people taking part, and we call these baseline characteristics. This makes sure that anything we think might change the answer to the question we're asking is collected. We can also use these answers to summarise everyone who takes part in our clinical trial and see whether things like their age, sex and ethnicity are similar to those of the wider group of people with the health condition that we're interested in. Next up is randomisation. So that the answer to our question aren't affected by things like people choosing to have one intervention than the other, no one chooses which intervention they get, and this is done using something called randomisation. Randomisation uses a computer to pick an intervention for each patient. We can also use this to make sure that our groups of people having the experimental intervention and control intervention are balanced for things that might affect the answer to our question, such as age. Now, once someone's been randomised, they can receive their intervention. We carefully check on everyone whilst they're having their intervention to make sure they're safe. Last up is data collection and results. So that we can answer our question, we collect a lot of information on people in the clinical trial whilst they're having their intervention. Once we've collected all our information, we compare it between the intervention groups. This tells us how the experimental intervention differs from the control intervention, and it's how we answer the question we started out with. We then publish this information in scientific journals and share it at conferences to share knowledge. We also use it to help inform how future patients could be treated and create new questions for the future. Now, that's how a trial works, but to make it work, we need lots of different groups of people. Starting in the middle, we need clinical team members, such as clinicians and research nurses. The initial idea for a clinical trial generally comes from the clinical perspective. And then once a clinical trial is up and running, we need clinical staff on the ground to carry out the trial and interact with those taking part. They are supported by trial managers and trial monitors. Trial managers make sure that everyone has all the information they need and monitor the overall progress of the trial. Whereas trial monitors visit sites to check that everything is okay and that information being collected on trial participants is being recorded properly at the ground level. The information collected on clinical trial participants by the clinical staff is collated by data managers, who check it on a wider scale and send queries back to the clinical staff. Statisticians, this is where I come in, then summarise all of the information and use maths to help to find the answer to the research question that the trial started off with. However, you can see that this jigsaw is going a bit across the slide. This is because there's one group that I've not mentioned. Patients. Without patients consenting to take part in clinical trials, they wouldn't exist. Alpha is one of the trials coordinated at Leeds Clinical Trials Unit and looks to help people with hand eczema. We're going to hear from Maria, who took part in the trial, and to start with, she's talking about how her condition was making her feel.
Eventually, I got a little bit fed up and I even a little bit depressed, and the GP referred me to a consultant. And through having her advice and support and being in a trial with her, we found that a lot of benefits came out of the trial that helped heal the hands. And as a result, doing the trial, I felt that I was helping other people as well, because they could use the results of the trial and the way that my hands healed, how quickly, how efficiently, how long were they healed for after the trial, that this information went on to help somebody else and obviously expand the trial.
So that's how Maria felt taking part in the trial. However, it's not just trial participation that patients and the public can get involved. Patient and public representatives can also be involved in the behind-the-scenes working of a clinical trial. They can be involved in the design stage, deciding whether a research question is of interest to patients and the public, and determining whether the patient pathway of the clinical trial is acceptable. They can also input into the application for the trial to be funded. Once the trial is funded, they can be involved in helping to write the information sheets I mentioned earlier, to make sure the information being given to potential trial participants is clear and concise. At the end of the trial, they can also input into how the final clinical trial results are fed back into both the research and patient and public communities. In addition, they can form a key part of overseeing the running of the trial, inputting into decisions to make sure the patient perspective is always considered. Outside of the groups formed by a clinical trial team, patient and public members sit on funding panels and ethical review committees so that patients have a say on whether research should be funded and whether it's appropriate. Hospitals and charities also often have patient groups, which hear and comment about potential and ongoing research. And now involvement in this case can either be a one-off experience or a longer-term commitment, and the individual themselves can choose what's best for them. Now, if you'd like some more information on some of the different things that I've touched on today, there are a number of different resources online. If you're interested in learning about what trials are currently recruiting patients, you can look on the Be Part of Research on the National Institute of Health Research website. If you're interested in how to be involved in research, but not necessarily take part as a research participant, you can look on the National Institute of Health Research website, I Want to Help with Research, or look on the People in Research website, which is a database where researchers advertise opportunities for patients and the public to be involved through small or substantial activities. If you're interested in finding a local patient or public group, you can ask your doctors or look on the charity's website. An internet search of the condition and location may also help to identify an existing group. Finally, at Leeds Clinical Trials Unit, we are developing a new public-facing website which will provide information about our trials and their results. There are already some trials which have their own website, for example, Alpha, and if you look on some of the charities' website, they will give summaries of different trials as well. So just to summarise, I hope I've given you an overview of what trials are, who helps make them happen, and how you could be involved in the future, not just through taking part. I just want to close with how I started. At the top of any trial is the aim to answer a question which might help to improve the healthcare of patients in the future. For example, the clinical trials run at Leeds Clinical Trials Unit have helped change how patients with blood cancer are treated and help to identify what different things might increase a patient's chance of developing a pressure ulcer. However, we can't reach that aim without the trial participants, patients, who give their time to support this kind of research. And without them, there'd be no clinical trials. Thank you for listening. I'm going to close my screen so I can see everything again.
Hello, that was fantastic. I really like the idea of lots of people coming together to make something a success. So the scientists, the statisticians, but actually also the volunteers and patients having such a role. That was really nice.
Yeah, trials and this kind of research is a completely collaborative effort. Like, we couldn't do it without the trial participants, but you need the different expertise as well behind the scenes to make things happen.
Yeah, that seems to be a recurring theme. Like Eileen talked about collaboration, you talked about collaboration. No pressure, Nick, you better talk about collaboration. We do have a couple questions in the chat, but I want to say we've got quite a few people watching. I reckon we can get a couple more questions in. Now, we do have another one from Jenny. She's being a star. So are the clinical trials run at Leeds for anything in particular? Are they focused on particular body parts, illnesses?
So the trials which we run at Leeds Clinical Trials Unit generally fall into 3 different divisions. So we have the cancer division, which focuses on cancer trials. Then we have the complex intervention division, which focuses on things like trials in care homes, elderly trials, and trials in mental health as well. And then we also have a new division which is called surgical interventions, devices and diagnostics, which is all looking into I was like, what's it again? And that's looking into like different surgery trials and different kinds of interventions and looking at how you can diagnose patients as well.
That's really cool. That's really interesting. So those like nursing home trials you mentioned, or care home trials, are those actually taking place in the care homes? Like, in my mind, you go to a clinic and you live there for a bit, or am I mistaken?
No, so the clinical trials that run in care homes, I'm working the cancer division, so I'm not an expert in that area, just to disclaimer. But yeah, the care home trials do take place in the care homes, so all the care home residents will be consented.
No, I like that. I like that a lot. I have another question. I'm going to be
Yep.
selfish. How long does the process take? There's a lot of phases there, and it sounds like quite an easy jump from one to the other, but the look on your face is maybe not.
It depends on the size of the trial and how complex it is. Some trials can take a very long time just to get up and running with the different approvals and things you need to get into place and to make sure you know exactly how trial participants are going to be treated. And then if you need to recruit a lot of patients, then you're going to need a longer time to recruit patients, and then you've got to collect the data on them, and then it's just all about the follow-up. So it does depend on the trial itself.
No, that makes a lot of sense. Well, kind of roughly, how long would you say, if you had to put a number on it?
Well, a trial which I'm currently analysing started off 10 years ago. So
Oh, wow.
it can take a
More
while.
then.
Not all that long, but they can take a while.
With the kind of more attention being paid in the media with kind of the COVID vaccine trials and things, are there more volunteers now? Are you finding that? Or I guess if it's patient specific, maybe not.
Yeah, I think the difference between the COVID vaccine trials is a lot of them were done on healthy participants, whereas in us, we build in our trials into the NHS. So we do recruit patients and look in improving their standard of care. So it's just a bit different in terms of recruitment.
Yeah, I see slightly different like demographics
Yeah.
there. That's really interesting though. Thank you so much for coming and chatting to all of us about it. It's really interesting. Maybe you'll have prompted some of the audience to sign up for trials. I'll let you go back to the green room behind the scenes, and I will bring up our final speaker, who is Nick. Please do join me on the stage. You might have seen him earlier. There was a video on Twitter, which I watched a brief amount of and then had to come to this. So Nick, I'll let you take it away.
Oh, thanks, Alex. So, hi everybody. I'm Nick. I'm a pathologist. I'm hoping just to spend a few minutes tonight to tell you a little bit about how we're working on applying artificial intelligence to help some of our clinical work. So you probably wonder, well, when you go in for a biopsy, or maybe you have a relative who will go in for surgery, someone is going to have to make a diagnosis. And actually, that's the job of pathologists. So I always find when I talk to people at bus stops or whatever, people will say, "Oh, you're a pathologist, that sounds really exciting." And of course, everyone's thinking of Silent Witness or Quincy or that sort of very exciting background. Actually, the reality for most pathologists is our job is very much focused on clinical diagnostics. So traditionally, we used to look at very thin sections of patient tissues. So these can be biopsies, like I said, or larger specimens. We look at them under the microscope, we make a diagnosis, perhaps even conduct a few more tests. And on the back of that, then the management is picked up by medical teams. So we don't really have any direct patient interaction at all. So we're kind of working in the background. The only thing that's changed for us over the last sort of 5-10 years, perhaps, is we've moved away from using microscopes to increasingly working with digitized images. So rather than having tiny little glass slides that we sort of fumble about with under a microscope, they get scanned on high resolution scanners, and then we can look at them on a screen and make our diagnoses that way. And that actually has been pivotal to being able to support a whole host of strands of research. So back to pathology. So how are we doing as a discipline? Actually, we're not doing all that well. And the problem with that is that we have globally an aging population and a growing population, which means that in terms of demands for clinical service, that's going to be increasing probably by about 60% by 2029. So that's huge. And actually, the problem that we have is there aren't enough pathologists to be able to carry on and pick up that workload. And that's simply because pathology isn't one of the sexiest areas, perhaps, for junior doctors to be able to work into. And so we've got this shortfall between a need and the provision of a diagnostic solution. And the figures I've put there for the number of active pathologists in the US just show how these figures are falling at the moment. And the same happens in the UK. There was a census done recently, and it shows that probably about only 3% of histopathology departments in the UK are adequately staffed to meet demand. So we thought we'd try and make use of all these images in digital pathology and see whether we can develop some solutions that should hopefully be able to ease workflow. And one of the models that we chose was breast cancer. So you probably wonder, why why breast cancer then? And unfortunately, breast cancer is very, very common. Women have a 1 in 8 lifetime risk of developing this. And the number of cases that we have in terms of new diagnoses each year exceeds 50,000, and about a fifth of those will result in deaths every year. And in part, that's due to the fact that we've got a very effective screening system, but also it provides a huge amount of cases that we need to review on a regular basis. And one of the things that we were particularly interested in is not just about diagnosing breast cancer, it's about the molecular testing that's done. So understanding that you have cancer is one thing, but actually also getting a handle on how best to treat it is another one. And in breast cancers, we typically do two things. One is we assess for hormone receptors, and we call them ER and PR. And patients who are expressing these receptors are then suitable to receive hormone therapy, so they can get some very effective targeted treatment. There are some other women whose tumors actually will be amplified for a particular gene called HER2. Don't worry about the details for that. But what that means is that we know that the tumors for these women will actually be responsive to treatment with a drug called Herceptin, which we've seen a lot in the news over the last few years. So it's important for us to be able to diagnose, but also to help select treatment. So what does breast cancer look like? And this is typically what we see as pathologists on a daily basis. On the sort of panel on the left, we've got benign samples. This is what stained breast tissue looks like. And there are a number of appearances there, but actually all of those are perfectly normal patterns. On the right, on the other hand, we've got malignant samples. And I don't think you need to be a pathologist to recognize that the appearance of the cells there is quite different. So it's all about pattern recognition. So where am I going with this? Well, there's some very interesting experiments done in Cambridge to see whether pigeons were able to come up with a similar degree of accuracy in terms of making diagnoses. And these birds were trained effectively to look at cancers and benign samples and peck on one side of the screen to come up with their own diagnosis. And if they got it right, as a reward, they got some corn. And actually, their degree of performance was pretty high. So what's this telling us? It's telling us that there is something about pattern recognition that certainly works across birds, it works across humans, and maybe there is a way that we can actually make use of that using computers to help us with the diagnostic process. And this is where the artificial intelligence comes in. So there's many, many different types. Artificial intelligence is huge, and a subset of that is machine learning and then neural networks. And this is the system where we are trying to recreate that pattern recognition that's done by the pigeons and by the pathologists. And one of the techniques that's most often used is deep learning. You may have seen that in the newspapers, and that's been very effective, certainly in ophthalmology. So this is looking at the back of the eye. And it's a very good technique to help with the identification of diabetes or glaucoma. It's worked also very, very well in radiology. And so we've been interested to see whether we can actually apply this to pathology images as well. And unfortunately, many of the attempts that have done so far don't work particularly well. And that's because the machine learning element to it is very much a black box. So these models learn to make a correct diagnosis through an iterative process, but the features that they use to recognize whether this may be a malignant specimen or a benign specimen are not really easy to trace. And a good example is an algorithm that was using this kind of technology that aims to try and differentiate between wolves. So we've got this sort of Siberian wolf sitting around on the snow at the top, from a husky, which perhaps to the untrained eye doesn't look massively different sitting at the bottom. And in fact, the algorithms perform quite well in being able to differentiate them. But what turned out to be the case, they weren't looking at the animal at all. They were just biasing their decision on the background. So it doesn't matter if it's a dog, it doesn't matter if it's a wolf. The fact that the background is snow makes it more likely to be a wolf, and the background being grass is more likely that it's going to be a dog. So we really don't want that kind of technology to be able to make decisions clinically. So we tried a completely different tack, and our approach was using statistical physics and computer vision. So this is sort of extracting features from the pathology images, and we tried to bring in an understanding of the underlying tumor biology. So we're sort of meshing three different types of data. And the nice thing about this technique is that it's white box. So in other words, we can lift the bonnet on it and understand how the decisions are being made, and it has a hard-coded algorithm at its core, which effectively makes these diagnoses. So in applying this to the breast setting, for those of you who don't know how the process works, I'll give you a very quick synopsis. So imagine you have a lump in your breast. You will obviously have this picked up by mammography perhaps, but you will have a biopsy done. And this is at the very left of this flow diagram. And then moving to the right, that'll be fixed, it'll be cut into thin sections, it'll be stained and either looked at under a microscope or scanned and looked at as an image. And then the pathologist who is looking at these images or slides will provide an initial report. But remember, we need this molecular testing, which will help us decide which is the best treatment for patients to be done. And so that's done using a technique called immunohistochemistry, or IHC. So we're looking at the brown staining in the images in the center. And unfortunately, that's not entirely reliable. In some cases, then need further validation using other techniques. And the take-home message here is it's quite a long-winded process. It's quite labor intensive, and all that time patients are waiting for their diagnosis. So we applied our system to it, and what can it do? Well, we were able to detect cancer, we were able to identify the grade of the cancer, so this is a measure of how aggressive a tumor looks, but we were also able to identify these hormone receptor and HER2 molecular subtypes. So that is just without using any of those additional tests, just using the original image and extracting that kind of information of it. And the beauty is it works on a par with the existing standard of care. So this is what's routinely done within the NHS. But it tells us other stuff as well. So it tells us about tumor infiltrating lymphocytes. And you're probably wondering, what are these? So in the image on the bottom right, that sort of general background is all cancer cells, and you can see a little constellation there of little blue dots. And these are cells from your immune system that have been recruited to clobber the cancer. And it's important to be able to measure these because actually that also helps in predicting how patients will respond to whatever treatment they get given. And then we can get other measures in terms of how variable the tumor is and how likely patients are going to respond to their therapy. So this is a huge step forward in terms of just the simple diagnostic angle. So if we go back to our original diagram, all that bit in the grey box, which involves that further level of testing, the original diagnosis, all of that can be completed within minutes by a machine. It creates a very simplified report for the pathologist who can then say, yes, I agree with it or not, and then sign it out. So what does it all really mean? Well, great, it sounds like we can replace pathologists, and of course we can't. This is just meant to be a tool to help pathologists in their workflow, to help increase their throughput, to help increase their accuracy, because effectively they have an algorithm that is providing them for every case that they look at with a second opinion. But the machine certainly can't replace the expertise that pathologists have in understanding the clinical background of patients. We've had a chat to patients as well, and they seem quite keen. This is great technology, but at the end of the day, they want clinicians to be able to make the decisions. So that's good. We're not out of a job yet. But really what's important is what the benefits to patients and healthcare services are. And as I mentioned, one of the things is accelerating the diagnostic turnaround time. And that's important for patients because that anxiety that you have whilst you're waiting for your diagnosis is gone. We can accelerate access to treatment, and obviously speed that up. But in terms of healthcare services, we're also cutting down the cost of not having to do all these additional tests. So how do we adopt this clinically? And just to keep Alex happy, it is very much a collaborative effort. So we need to test. It's great doing this in the lab and doing this in the sort of ivory tower of academia. What we really want to know is, does that work across different settings, across different hospitals? And going back to what Kara-Louise was talking about, we need to do clinical trials. So this is where we're at the moment, is validating this across different hospitals in a collaborative manner to see how robust it is. So what are the wider applications? Well, I just want to leave you with the fact that essentially this is computer-based. So this is a technology that is very easy to set up in the cloud, which means that you can access this kind of support service from wherever you are in the world. It can standardize the way we report histopathology, so cancers. And of course, in places like Rwanda or Burundi, where you have single figures of the number of pathologists relative to populations of 11, 12 million people, actually they don't have access to pathology services at all. So something like this could actually provide that bridge until they can get their healthcare services up and running. It relies, of course, on having a digital system, but that's fine. That's affordable for most economies. And what we've been doing more recently is we've been moving this outside the breast cancer space. And we know that it works for lung cancer. We know it works for skin cancers. And quite excitingly, recently, we've seen that it works also for large volume specimens. So this is the sort of cervical smears women go for on a regular basis. And so we can provide that additional input and hopefully change the way that we're going to be reporting cases in the future. So thank you very much. I'm just happy to take any questions.
Nick, fantastic. Thank you so much for linking together into the collaborative side of things. We were just saying in our own personal chat how well everyone's linked together. Perfectly planned, I would say. I think my main takeaway from that was I shouldn't be scared of robots stealing my job. I should be scared of pigeons stealing my job and potentially doing a better job. You win some, you lose some. We have got quite a few questions in the chat that I'll be generous at the end and I'll let them go first. So we have another one from Jenny saying that she found this really interesting and wonders, is this technology going to help you select better treatment plans for patients?
Yeah, absolutely. So this is exactly where we're trying to go with this. As a discipline in pathology, our bread and butter has been diagnosis and trying to inform oncologists who deal with cancer treatment as to what the better options are. But there are areas where we have complete unknowns at the moment. We can't predict with very good accuracy which patients will respond particularly well to a given kind of therapy. And by extracting that kind of biological information from these images, from these tumors, we can actually refine that degree of prediction of how patients are going to do, and therefore try and avoid some therapies which we may have been giving patients otherwise and may not have benefited from. But don't forget, chemotherapy is very toxic, and unfortunately it has huge side effects. If there's no benefit and we can avoid giving it to specific subgroups of patients, we should do.
No, that makes complete sense. And that actually links really nicely to, I'm very sorry if I'm about to butcher this name, Olwen, because he says, presumably since it's a white box technology, as you learn more about cancers and supposedly as the AI kind of tells you more about cancers, you can then update things and that might help with these unknowns.
Yeah, so that's an excellent question, actually. We could talk about it for days. The algorithm that we have is fixed at the moment and it works very, very robustly. What we do in terms of improving it is in how it deals with the images themselves. So different tumors from different organs have got very, very different cellular backgrounds, and it's how the algorithm tackles this variety that needs to be optimized for every organ. So yes, we do improvements on a regular basis in that regard.
Oh, that's really good. It's good to know it's getting updated, even if the algorithm at the moment is kind of fixed. I have a question actually from the people moderating in the comments. So now things just take minutes to diagnose. How much time are we saving here? How long did it take before you kind of had this automation?
It depends how good your service is. In terms of, so it varies across different hospitals and obviously across different countries, but even in the fastest sort of setting, it may take you a couple of days with a breast biopsy to come up with, a complete diagnosis, and we can shorten that process considerably.
No, that's good to know. Days versus minutes, yeah, easy decision there. So getting the treatment earlier, I suppose, does that then have direct impacts on survival, or is it kind of too soon to know that just yet because of the clinical trials?
So we're very lucky in the UK actually at the moment that our cancer care pathway is pretty much streamlined. So patients do get their treatment very, very quickly. But this is the UK. And even if you go to other developing economies, like developed economies like the US, things are very, very different. So they could have certainly a benefit in that respect. And then if you go to places like sub-Saharan Africa, they have nothing. So something like this could be a really affordable way of accessing cancer treatment.
Right, and I imagine because it's kind of the scanned images of slides, you don't even need kind of trained professionals. You could have the biopsy, have the slide, have the scanning, and then get that sent over to somewhere that maybe has the technology to then run the algorithm.
That's
Really
exactly
cool.
So just the tissue processing you could do in a field lab if you wanted to.
No, that's so cool. We do actually have a question as well from Shawnee, which is incredibly on topic. Apparently, pathology isn't the sexiest medical area. What are the better ones?
Well, I disagree. I'm a pathologist. Yes, a lot of people, I think, start out without fully knowing what they want to do. Lots of people go into surgery, lots of people go into medicine. I think the reality is a lot of people end up as GPs, and there is a huge need for GPs at the moment as well. I think what people miss in pathology is we don't interact with patients at all. And it's a big, big change, because as part of your clinical training, you work with patients, and from one day to the next, it's you in a computer or you in a microscope.
Yeah, I can imagine. Actually, it's interesting what you said about kind of the patients want clinicians, not robots. But it sounds like you're not even there with the people anyway, so you kind of, in that case, are a robot in a way. Not the conclusion I meant to have from this. No, that was absolutely fantastic, Nick. I definitely learned a lot. Yeah, thank you so much. So please do join me in thanking everybody that spoke to you tonight in the chat. And I'll bring tonight's session to a close with a nice 15 minutes for you to get ready for the football. So I'd like to thank you all, thank all our brilliant speakers. That's Eileen, Kara-Louise and Nick. I had a really fantastic time, and I hope you did too. Judging by all the comments in the chats, it sounds like we did. If you did like this event, please do let us know by tweeting us at Uni Leeds Engage and using the hashtag BeCurious21. And we really want to get some feedback on tonight's event. So, maybe you want to say all the good things, like the hosting was fantastic, or maybe there's things you didn't like so much, like we should have more Alex or more pigeons doing this. And our moderators will also share this link in the chat for you, so you don't have to copy it down. And if you've booked onto this event, you'll get an email tomorrow with the link. So don't forget to have a look at the rest of the Be Curious programme, which is available at www.leedsac.uk /Be Curious. All the events are free to attend, and I would absolutely love to see you there. So that's it from us. Enjoy the football. Fingers crossed it comes home, and goodbye.